Yeah.
Brilliant. Okay, so thanks everyone for joining. It's an honor to have Chensu joining us today for our TIA Centre seminar. So Chensu joined in 2016 the Weill Cornell Graduate School of Medicine and Medical Sciences to pursue a doctorate in computational biology and medicine. And since then, he's joined the computational pathology group at Memorial Sloan Kettering Cancer Center with Dr. Thomas Fuchs. So one of his highlights includes developing a deep learning model for nuclei detection, and he's been recently involved in work extending this model and applying it to various downstream applications. So it's an honor to have you today here, Chensu, to present your work. And yeah, for the people joining the call, please feel free to answer or to ask any questions as we go. And Chensu's agreed to field those during the presentation. So I think with that, it's an honor to have you here, Chensu, and whenever you're ready, please share your screen and start presenting.
Okay, I have access now. Okay, good. Thanks for the nice introduction. Yeah, hi everyone. I'm Chensu Xie. I've been working with Dr. Fuchs in MSK for a few years and just graduated recently. And yeah, thanks for the invitation to share my recent work on this computational biomarker, which we developed to predict the patient response to immunotherapy in lung, which use lung immune checkpoint inhibitors. So this is an overview. We basically use a way by looking at the cell interactions to identify infiltration hotspots. So the regions that we think potentially have immune events happening. And we extract those tiles, and then we map from all of these tiles to the outcome, which in this case is the response. So yeah, talking about computational pathology, we have a lot of goals. In my mind, I think the ultimate goals for this field is to map from the patient image slides and also maybe combine other multi-modality data to the patient outcome. We have seen a lot of work on diagnosis, including metastasis analysis, increasing grading, representations, and also basic detection of cancer, classification of tissue types. And also we are looking, we also saw some works on genetic mutation prediction from whole slides, and also prognosis, of course. And our work here is focused on the treatment response prediction. So yeah, so just giving you an overview of how this immunotherapy is working. So basically, there's a suppression system. We call it immune checkpoint. So it's basically a receptor binding between T cells and tumor cells. And there's a ligand expressed by the T cell called PD-1, and on PD-L1, which is this ligand, expressed on the membrane of the tumor cell, and their binding is actually working as a suppression system to the T cell. So if we can, suppress this suppression, or we can break this binding or whatever, this suppression system will be damaged, so that our immune system can work better against the tumor cells. So that's the idea behind this treatment. And there have been many types of drugs being used for these immune check. They are called ICI, which is immune checkpoint inhibitors. They can be targeting at PD-1 or PD-L1, which is basically what we're looking at in this paper. And there are also other drugs looking at CTLA-4, and one way of measuring the response to this immunotherapy treatment is to look at, the following CT scans of the patient, comparing to the CT scan before the treatment. So you may basically measure the size of the area of the nodule, and you have a proportion of shrinkage or even expansion or stable disease. So this proportion of the area or size change of these nodules gives you a number, and based on this number, pathologists will classify these patients into basically 4 categories. They call them complete response, partial response, and also progressive disease. That means you see expansion of the size, and also stable disease means that there's basically no response, and but not getting worse in terms of the size. So we classify these 4 categories into 2 classes. So in which complete response and partial response are positive, which is responders, and the other 2 are non-responders. And the challenge of mapping from tissue slides to the patient outcomes, not only response prediction but also prognosis, is that there's a gap of knowledge between understanding the tissue slides to the patient outcome. And this gap of knowledge basically creates bottleneck of supervision at tile level. So if we think about the diagnosis, it's pretty straightforward that people want to look at the tumor regions, or you give a tile level annotation of tumor regions, and you give them a label. If it's just the detection, you can give 1 and 0, 1 to the tumor regions, or it might be many types of cancers. You can give them multiple labels, but where to look at and what kind of features is clear here. So you're basically looking at the morphological features of the tumor regions. So for prognosis and response prediction, we're not lucky, and so therefore we have to do more explorative modeling, which we have to let the model itself or other ways to identify the location, nature. Peri-tumor features. I don't really understand this term, sorry.
So basically that means features related to the surroundings of the tumor, not the core of the tumor.
Yeah, for diagnosis, I think I'm not extremely expert in all of the features that may be relevant to diagnosis, but I think to date, most of the models working on diagnosis basically looking at the tumor regions, and for grading, or at least for detection, I think if you have annotation, basically people are just giving you tumor regions, and you just need to do tile level classification. Or if you do end-to-end, you can do a mill, but in the end, you are just detecting the tumor regions. But if you're talking about other clinical outcomes, we do find that these so-called peri-tumor features might be relevant, because some of I was showing to my group, to our group, that sometimes the stromal regions can be of some kind of significance in terms of the response prediction. But I think, there's no established idea how this information should be integrated and how they are relevant to different kind of clinical outcomes, which is part of what we have to explore. But I agree, we should definitely not only focus on tumor regions, and that's why there should be more end-to-end learning on the whole slides rather than annotating the tumor regions. So
I guess if I can just maybe make a comment, Chensu. So
Mhm.
I assume that sometimes when you're analyzing the tumor regions, maybe you're doing maybe an initial coarse localization. So you are potentially still actually considering some of those intertumoral areas if you're doing that more loose localization at the start. Do you agree?
Yeah, it depends on the way you do the analysis, right? If you're doing tile level, you are looking at the tile, which maybe it's within the tumor core, but you have like a tumor, like intratumor stroma regions,
Yeah, exactly.
something like that. But what I'm talking about is a more general idea. You know, for a diagnosis, you want to look at the tumor regions, which might include the stroma and also blah, whatever. But the algorithm is looking at these regions. So that gives us idea. So we try to look at the tumor regions only for response prediction, and also for prognosis. For prognosis, is working pretty fine. And later we added the surrounding regions. Also, I mean the whole slides, not only the tumor regions. It has a very minor improvement. But for response prediction, we tried a lot of things and finally converged to very selective regions, which are we call infiltration hotspots. So the two-stage approaches for whole slide assessment is pretty much a standard now. We have the tile encoder, then we have aggregation. So most of the because the features or the predictive information is actually carried at the cellular level, cell level. So you can't just use a low resolution image or thumbnail image for the prediction. So people have to look at the details of the tiles, and they encode these tiles into latent space, which is a lower dimension, then do aggregation. This aggregation can be aggregation of scores or aggregation of the tile features, then output the slide level. And just giving you two examples. One is the multiple instance learning, which applies to tumor detection. Basically, it has an assumption that single tiles, not necessarily a single tile, but single tiles can carry enough information for the whole slide classification. So you can regard this slide as multiple instances because a single instances is already, the label for the single instances equals to the slide label. And for unsupervised theta E training, this paper give us an example of a more complicated task. They want to predict the survival because the Gleason grading has been the standard for survival analysis for prognosis in many years for prostate cancer and for other cancer types. So people believe that clustering of the tiles, like tumor subtyping, is the key to prognosis analysis. So this paper basically do a clustering of tiles. They have an unsupervised training of the tile encoder, and then they cluster the tile features and select clusters, important clusters for aggregation. And these two methods given us some idea how to do the two-stage approach, but there are some problems. For example, for unsupervised theta E training, you are not really optimizing the tile encoder against the slide label, so you cannot guarantee that the tile features you learned can be predictive, especially in the case for outcome prediction, like response prediction. It's pretty hard to say what kind of feature is important. And for multiple instance learning, the relationship between the tile and the slide label is pretty simple.
Can you see that, Chensu?
Okay.
I
I don't know how to turn on the.
Are you on PowerPoint? If you're on PowerPoint, at the bottom left, there should be three dots, and then you can hopefully, yeah, there you go.
Yeah,
perfect.
Like this.
Nice.
So for multiple instance learning, we have the assumption that there's a clear relationship between the tau level and the slide level, but that's also not the case for response prediction. So these are a summarization of the points that I just mentioned. So to solve this problem, and we want to develop a new model. So first, if you want to do, if you want to look at very diverse information of the whole slide, and you make a good reasoning on the tau features for the outcome prediction, you ideally want to map from all of the taus, not only in the tumor regions, not only in the stroma regions, because you don't know where the useful taus are. So you want to map from all of the taus to the slide level. But this is not feasible because of the limitation of, for example, GPU memory. And so we propose to train a model, look at model the whole slide as clusters of tiles, so of similar features, because the same motivation as the prognosis. So if you look at the whole, if you regard the whole slide as a group of clusters, and each cluster represents a subtype of the tissue, and then you use every centroid. So these three circles here as centroid, which is the average of the tile features of the clusters. You want to, in this way, reduce the model to a smaller computing graph, because you are mapping from the whole slide, from the slide level going back to the centroids, but you still have to calculate the centroid by averaging the tile features. So therefore, we further do a relaxation. Instead of calculating the centroids, we choose a tile of every cluster to represent the centroid. And that tile is chosen based on the Euclidean distance in the feature space. So we basically choose from the nearest tiles to the centroid, and use this tile as the so-called centroid approximation tile to represent the tissue subtype. So in this case, we can train the tile encoder from end to end, and also the aggregation layer. So this is an overview illustration of this method. The image is tiled into small patches, and these patches are encoded by the tile encoder to the feature space, and they form clusters along the training. And these tiles that are used to approximate the centroid is then extracted. These features will be concatenated and feed into the aggregation layer. So this aggregation layer is connected to the L times K, which is the number of clusters. L is the feature length. This concatenated feature vector, and this concatenated feature vector is connected to the tau encoder. So this is basically trained from end to end, looking at the tissue subtypes. So it's mapping from tissue subtypes to slide level, and in this way, you can train the tau encoder, which is very critical in this whole step, against the slide level. And in the meantime, we want to have good clusters. Therefore, we want to push the tiles to the centroids in feature space. And what kind of tiles, what kind of features or clusters can be learned is totally depend on the data and the model itself. So here is, sorry.
If I can just ask a question. So first of all, how do you determine the number of clusters that you're going to use?
This is quite empirical. There's also a limitation of GPU memory, right? You can't use a very large batch size and also a very large number of clusters, because you are training from end to end, basically. But it's a trade-off between memory and the speed. You can do a method called checkpoint in PyTorch, so that you can have theoretically infinite number of clusters. But if you want to train faster, you want to make copies of tau encoder in the memory at the same time, and do the activation, do the optimization directly from end to end. So and also if you choose too few number of clusters, the model will not perform well. I had a comparison between cluster number of 1 to 2, 4, 8, and only until 8 you can see better performance. The idea is that sometimes, especially for if you are looking at this model for diagnostic problems, or so you're looking at the tumor tiles, but there are stroma tiles, for example. You cannot really categorize the stroma tiles into the same cluster as tumor tiles. It's a very hard objective for the model to do that. So because they naturally form some patterns, the tissue tiles. So if you use too few number of clusters, it's going to be giving you some issue. If it's too many, I have no idea what's going to happen because I haven't really tried.
Okay, sure. So I remember your previous slide, you said that ideally you would like to feed all the slides into computer memory, but you just haven't got the capability to do that. So I guess you're trying to balance what you can fit into memory.
Yeah,
okay,
exactly.
Question, if that's okay. So Dang online said, have you done any clinical verification of these centroids?
Clinical verification means presenting the centroid approximation tiles to the pathologist to look at what they are.
Yeah, exactly.
We have some model introspection. Look at the not only the approximation tiles, but also the nearest tiles, like 20 tiles nearest to each centroid of the whole cohort, to look at what the centroids represent. And in different cases, they represent different things. Sometimes you can recognize what they are. For example, this model was trained against cancer classification. You can clearly see in the most important cluster will be tumor-like tiles. For positive tiles, you have positive slides. You will have tumor tiles in those cluster identified. For negative slides, you usually don't have any tile belonging to that cluster, or you have a tumor-like but benign tile staying in that cluster. The reason I'm not showing here is that the focus is not really classification here. And for survival and treatment, for survival, it shows completely different pattern, and we couldn't really recognize. We have some description, but not fully describing every cluster in this case. To establish a domain knowledge require a training of decades for the pathologist, and you may have to present as many tiles as the model is looking at to the pathologist, so that they can actually give you some idea what's happening behind this. But in most cases, it's pretty hard task, even for experts, to look at the tiles and give you a description, which can convince themselves.
Okay, sure. And we have one more question in the chat, if that's okay. So Mustafa would like to know a bit more about how you aggregate your concatenated features. Is this a neural network?
Mm-hmm.
So theta A, basically.
Theta A. Also, this is a framework for detection. Theta A here is just a single layer, fully connected layer. And for prognosis, we have MLP, multiple layers. It depends on. There are other possibilities, and I have tried using compact bilinear pooling when I have a lower number of clusters. And you can also look at higher order interactions between these tile features or tissue subtype features, or maybe you can even try self-attention pooling, something like that.
Okay, brilliant. Thank you.
Okay. So thanks. I'll go through this very quickly. So for the whole cohort, all of the tiles, all of the slides, and map through the tau encoder, which is initialized as random. So you have tau features randomly, and then you assign these taus to you assign a cluster label to these taus, also randomly. Now you have very bad clusters in the feature space, but you have clusters. And for for a single slide, you have a subset of the taus in a whole cohort, which is represented by these dashed regions. And you have the slide-specific centroids, which represent a shifted tissue feature. And these taus used to approximate the subtype feature will then be combined, fit through the tau encoder and then combined to the aggregation layer. And also this training includes another loss pushing the taus towards the centroids, and they are trained concurrently. And we have part reassignment, because after a while of training, the theta is being modified during the training, and in this way, it might no longer be a good approximation from the tile to the centroid. So we have to actually averaging the new features of these tiles belong to the same cluster to get the new tile centroids, and then we recalculate the new approximation tiles. So this iteration goes on and on after every epoch. And in this way, you can regard this algorithm as a elongated k-means. Yeah, this is a general framework, and it allows for explorative modeling with path learning. And in the response treatment prediction, we actually replace this tau encoder to a graph neural network. And I'll be going to that very soon. So back to the clinical outcome prediction. In this case, is the ICI treatment response prediction. We actually collected a quite big data set, which we have 583 patients in this case. We're mapping from the H&E slides to the RECIST criteria, the criteria I mentioned based on the following CT scans. So this is called the RECIST. And there are other groups, other overworks mapping from, for example, genomics or multimodal data, or from H&E to the survival. So, and also PD-L1 expression has been approved by FDA. So mapping from PD-L1 expression to the survival, both PFS and also OS is approved by FDA as the biomarker for the response for the drug. And there's also estimation. So there are different works, this is a manual estimation of PD-L1 expression from IHC stain slides, but also people use computational models to replace this or to standardize this PD-L1 expression estimation. And also there are estimation works mapping from the CT scans, following up CT scans to RECIST. So these are all just standardizing these manual steps. So because RECIST it is effective at splitting patient groups. So we basically group the patients based on the RECIST outcome. So RECIST-based responder and a RECIST-based non-responder. And it seems that both OS and PFS show a very good patient group splitting. And also why we choose RECIST as the training label, because we think from a machine learning perspective, it should serve as a better training model, training label. It should be cleaner because between the RECIST response measure to the death or PFS, we have maybe other kind of treatments, progression of disease. So there are many variables that can affect more than the slide itself. So we choose RECIST as the training label, and this is the overall workflow that we finally converge to this model. So we basically look at the whole slide, detect all of the tumor cells and lymphocytes, and then we look at their interactions spatially. And then we do this so-called infiltration analysis so that we can have the infiltration profile across the whole slide. We can look at which space have greater intersection between these two cell types. And then we extract these regions for analysis. For this analysis, we are using a graph neural network as the encoder. Extract the node features, basically the cell features, by self-observation. And also the slide infiltration scores. The global feature of the slide will also be concatenated to this model. And finally, we output the response. And we use this model to do the cell nuclei detection. Basically, this is a very old paper already. Just show you an idea that we want to look at a very accurate location of the cells. So instead of doing segmentation, we want a coordinates as accurate as possible based on the annotation of the pathologist. But anyway, pathologist annotation may have some, inconsistencies inter observers, but we had tried two pathologists annotating the same tiles, and the location variation is quite small. So it seems that they have a pretty good agreement where the center of the cell nuclei are. And after these are the sample patches of the lymphocytes and tumor cells we detected, because we have no quantitative way to measure the performance over the millions of cells detected. We show these lymphocytes to our pathologist and also these tumor cells, and we did a hierarchical clustering of these cell types, and it seems satisfying. We are actually detecting the lymphocytes and tumor cells, and then after we done the detection, we identify the lymphocyte spatial clusters and also tumor spatial clusters by an algorithm called the DB scan, which basically is density-based clustering. We grow the clusters like a greedy algorithm. We grow the clusters based on the distance between the cells. So we have this spatial interaction between lymphocytes and tumor cells, and we regard this as potential immune events happening. So we call these positions as infiltration hotspots. And we have this heat map of the whole slide. Then finally, we extract tiles based on this heat map. And you can see this is an individual case of study. And in the middle is this case of standard care. It has H&E stand. And we use this model to look at the infiltration hotspots and extract the tiles, combining with the global slide features and output the outcome. And in this case, the PD-L1 expression seems to be completely negative, but this actually is a responder, the ground truth. So, sorry, it's a question.
Yeah, if I can just ask a question. So I'm trying to understand, and maybe people on the call have also the same question in their head. So before you mentioned that you have the detections, right? So you have the tumor and the lymphocytes detected. So you said that you performed some spatial clustering, but also you performed some hierarchical clustering. So can you explain what here is and why the hierarchical clustering was that just for visual confirmation, or was this using
Yeah, so the hierarchical clustering based on the cell features is just a visual understanding of what we detected. Okay. And the spatial clustering is to analyze the spatial interaction or infiltration of these lymphocytes and tumor cells. So that's the point of feature clustering there and spatial clustering here.
Yep. Okay, makes sense. So the hierarchical clustering isn't used for downstream analysis, just for
No,
the
just
confirmation.
for visualization of, we can just randomly select cells to present to. We want a more systematic view of what we detected.
Yeah. Okay, makes sense. Thank you.
Yeah, this is the similar model, but the only difference is that we will replace the tau encoder to a graph neural network. In this case, we use graph isomorphism network to look at the cell interaction patterns and also cell features, and then these graph features are clustered in the feature space. And the other difference is that we also include the infiltration scores, which describe the level of intersection between the lymphocytes and tumor cells. And we have 2 scores concatenated to the slide feature vector, and then we output the response. So, and this is also showing what kind of regions. So interestingly, they are forming different regional patterns of different clusters, and we have shown these tiles with, in this case, every tile we presented, you can see the dots here within the tiles. These are the cell centroids, cell nuclei detected, and we color it by the node importance, which is another level of model introspection. And the red cells seem to be the most important to the classification. So, but we couldn't really identify a meaningful description of these kind of cells, how they are different from the other cells in the end. And yeah.
So if asking so many questions here, you actually might be about to explain it on the slide, but there's various ways to actually measure the node importance, right? So with GNNs, you can use GNN explainer, or you can have graph-based alternatives to, let's say, for example, GradCam, or you could even have like integrated gradients. There's quite a few different methods. So I was just wondering what the actual method that you use to denote the node importance was. But it seems like you're about to explain it on the next slide.
Yeah.
Okay, thank you.
Yeah. So yeah, and basically, I was going to talk about the GNN encoder architecture here. I will not go into very detail, but it's basically a 4-layer GNN, and each layer is a graph isomorphism network layer, GIN layer, and followed by a top-K pooling. Top-K pooling, basically, you learn a vector which is the same size, the same dimension, and that vector times the node feature, and then gives you a dot product, which is basically the importance score. And in this case, you basically you filter out the node that's below some threshold of the importance score, and layer by layer you have less and less nodes features recorded or actually outputted, and then you average. We basically actually sum the node features after every layer, and then we concatenate these node features, these sum node features, to be the final graph feature of the tile. So this is the final feature of the tile. And the importance calculated based on the top-K pooling score learned. So you have the importance score of the node, and also an estimation of the layer importance. So this is basically a weighted average of the node importance times the layer attribution, the layer importance. So that's how the importance is calculated. And this is the quantitative results we have shown. It's basically comparable performance to the PD-L1 biomarker, which is FDA-approved. But it is known that PD-L1 is missing a lot of responders. So in this case, we also observe this kind of scenario. So there are many patients that are actually responders, but they have very low PD-L1 expression level estimated. So they may have 0 or even like 20%. You know, the estimation, the manual estimation from pathologists are a percentage score. So like 20%, 0%, 5%. These patients are actually responders. And in this case, the model is correctly identifying them as responders. It has a much higher sensitivity, while it has a similar performance. That means the specificity is a bit lower than the PD-1 expression. But other than that, it also has many other advantages. You know, PD-1 expression level estimation by IHC, you need an extra staining, and you have more technical costs. You have a turnaround time of 2 days. But just working on H&E itself is pretty much a standardized protocol across institutions. And also we can avoid in the observer variation, and the technical cost is low and time is fast. And finally, here is the model introspection. So first, the thing is, if you look at the top left, we have the tau importance recorded or estimated by the gradient. So I have that discussed in detail in the EPO paper, but here, just to show you the results, we have the importance of different taus and rank them from top to bottom. And these top two clusters or parts seems to be the most important for the classification. And on the left here, you see every column here is a patient, and each tau here is the central approximation tau that the model is looking at. So you basically see some of the slides actually missing a lot of taus. They have very different size. Some of them have a very large amount of tissue. Some of them are very small biopsies, needle biopsies. And anyway, the top two rows, top two clusters seems to be important. And one is positively correlated to response, and the other one is negatively correlating. And we have shown this cell importance as well. It allows us to make more discoveries in a later study. And we can also map this tau importance score back onto the in situ mapping back onto the original slides. You can see where the taus are extracted from and where the important taus are extracted from. Here's just a statistical presentation of the difference between responders and non-responders. You can see non-responders, the cluster 1 and cluster 2 seems to be quite correlating to the patient outcome. So I actually had another experiment. I basically used the proportion of cluster 1 and cluster 2 as the single score, the single metric for classifying responders versus non-responders. It can also reach 0.6 AUC, which is interesting. And here, finally, I want to mention some previous efforts. We didn't just come up with this model from nothing. We have tried many hand-engineered features. So that's the first thing we thought about, because there's a minimal theory about what kind of features can be predicted for the response. But one thing known is that the drug is targeting at the immune checkpoint. So it's targeting the PD-L1 and PD-L1. So looking at the cell interaction between lymphocytes and tumor cells might be a good idea. So that's how we decide to engineer features based on the cell nuclei detection, based on the distribution, distance, many type of spatial statistics. And then we found there are two scores that can be showing us some signal of the treatment response prediction. So these two scores are finally also included in the graph model. as the slide features concatenated to the outputs of the neural network. And we also tried very simple baselines. We do a pseudo label, given the pseudo label, which is basically a slide label to every tile to do the classification. We also tried neo. All of this method really doesn't give us a very good result. So that's why we conclude that maybe single tiles cannot carry enough information and let us know whether the patient will respond or not. And in this case, we finally saw that there are two clusters. One is positively, one is negatively corresponding. So there might be more relationship or more higher order interaction between different tissue subtypes that waiting to be explored to improve the performance. Yeah. Question?
Hi, Chensu. So just a question about the comparative method that you used for MIL here. So which approach did you use to compare?
So it's the top. It's not attention. We kind of vanilla, but not the top one. The top K tiles are used to averaged to represent the score of the slide.
Okay, makes sense.
Yeah. So if you talk about male attention, you're doing a weighted average, and our model has a kind of a feature interaction, but still there are more to explore on this end, especially if we have a tile encoder trained in this way. It's already a trained tile encoder. It can make some sense. Maybe we can explore more later. So yeah, back to the slides. So yeah, just a quick conclusion that if we decide to use a model which can map from end to end and analyze complicated tasks like response prediction, it might not only predicting clinical outcomes, which is basically what we want to do in the field of computational pathology. It might also, in return, establish new domain knowledge. Although this is pretty hard, even if you do model introspection, you show the tiles that you detected, and also the clusters, how the tiles, what kind of tiles are grouped together. Still, it's pretty hard for the pathologist to understand what the model is looking at, because anyway, the model is looking at has already looked at thousands of tiles, and it's really hard to convince somebody to go through the same process of the model. And yeah, so to conclude, this is the lab photo of our lab. Although now there's only Thomas and the Chao Feng, and I also just graduated. And this lab is almost now is just two people. Yeah, thanks for attending this presentation. Yep.
Great. Thank you so much, Chensu. That was a fantastic talk. Really interesting. I loved how you have the when you're mapping it back onto the whole slide, you've got the tile level information and also the node level, the nuclei level information. So that's really interesting. Saad, you have a question?
Hi, Chensu. So I have a question. Something very basic first. So basically, do you do any kind of, most probably you do some kind of stain normalization up front, right? Yeah. So that's, and like, how is that reference target, etc., selected? The other question being that, we've seen with histocartography, for example, that what kind of distance thresholding that you use for k-nearest neighbor to compute the graph also kind of throws things off quite a bit when you're doing, tumor subtype classification or anything like that. So was that also empirically set the distance thresholding to create the graph before you throw it into GNNs?
Sorry, I didn't quite get the second question, but the first question is about the normalization of the staining. I didn't do any normalization of the staining
at all. So
at all. Yeah.
all right, that's quite interesting. So and then you had an independent test set, which was from a different institution, or
no, that unfortunately it's very hard to get this kind of data set of results. So I showed. So the test set is also from MSK.
all right.
But yeah, it's including this number. You know, the resist measurement is pretty it's pretty hard data to get.
Yeah, sounds good. So the other part I quickly want to mention was that, you know, for all these H&E slides, obviously you will have IHCs images too, with different expressions for PD-L1, maybe some other kind of markers too. Rather than jumping the gun and, you know, going directly for H&E, there is also a possibility that you start off from IHC, right, where you can establish the domain knowledge and the true protein expression directly, and then kind of relate back to the domain knowledge, or, you know, true biology. Whereas if you start from H&E, obviously, then you are leaving a lot to pathologists' perception of, you know, what they can figure out from their textbook knowledge or not.
Yeah, you mean mapping from the IHC, PD-L1 stained IHC slides to the outcome.
Also, there might be like some other kind of protein expressions for the IHCs too, right? Like not just PD-L1, there might be CD3, CD8, and FOXP3, depending on the clinical protocol. Then that allows you to naturally just take that into account even for tumor infiltration analysis, rather than actually, you know, creating even a deep learning model to do that. And that gives you a much truer information.
I agree. Actually, it's very it's a very good point. But one thing that one of the reasons that I didn't try different staining is that because it's, you know, H&E, you can easily obtain this for every patient, and it's standardized. And IHC is, you know, even for these cohorts, we have many patients. We have only like 70% of the patient has the IHC measurements, and also PD-L1 IHC measurements. So it's not easy to get that data, first of all, and also IHC staining is not as a standardized protocol as H&E. It has more variations. You know, there are many studies talking about the variation of staining of IHC, but that's actually a very good idea, and that's one thing that we should definitely try in the future, looking at a different channel of the tissue slide images.
I think also this is part of the power of potentially this approach, because you can uncover subvisual information and try and relate that to, for example, the response. So I guess with IHC, that's been used in the past because pathologists can easily visualize what's going on and see what biomarkers are actually of interest. But H&E it's a bit less clear, like you mentioned in your previous slides. So I guess you're trying to uncover those subvisual patterns, aren't you?
Yeah.
Great. Has anybody else got any questions? Anybody in the room? So maybe I can ask another question. So of course, in one of your first steps, you detected different nuclei, and then you determined the hotspots and extracted some tiles. So for each tile, you had your nuclei fed into a GNN. So can you explain what those node level features were that you used in the GNN?
So the node features are is trained in another model. So it's trained on another cohort, which is a 10 class, 100 slides from each type of cancer. We obtained 10 times 100 slides, and we extract the we extract the cell features on this, and extract the cell coordinates, and then we basically by replacing the background, also contrastive predictive learning, this kind of self-observation to train to get the cell nuclei features. So these cell nuclei features based on 64 by 64 patch size, and these will also be extracted in this case for all the lymphocytes and tumor cells. We didn't detect other type of cells, but there are false positives, which is a very small portion of stroma cell is also detected to be lymphocytes in some cases. But in any way, in the end, we didn't use the class label. We used the node features. So node feature representing every nuclei, and also the topology to the graph neural network.
Yeah. So you use mainly the deep features. So I wonder whether there's still a bit of a so for example, if you've got your various nodes that you flagged up as important, I wonder whether it's still quite hard to actually interpret why those nodes are important. What do you think about
that? Yeah, I try to convince my collaborator to look at as many tiles as possible, and I also try to show them only the cell node feature importance, and also show the graph as well, maybe. But it seems that maybe I haven't shown them enough. But anyway, I think it's pretty hard to understand what the graph neural network is looking at, because you are looking at the summarization of the cell features, and also, a summarization of the cell features according to the topology, and then also another layer of transformation, nonlinear layers. So it's pretty hard to tell why the model thinks this cell is important. And from the perspective of pathologists, they are not important. They are not that different from the other cells around them.
Okay, brilliant.
Yeah.
So Saad, we unfortunately haven't got time for your final question because there's somebody else who needs to come in and use the room. So if you would like to, I'm sure you can reach out to Chensu and ask that question. But thank you so much, Chensu. That was a really exciting presentation. Yeah, so looking forward to seeing more of your work in the future.
Thank you. Thank you.
Thanks so much.
Thanks.
Bye.